ICGB is an international network of different countries, where different countries have formed a strategy to develop biotechnology research in developing countries and to utilize the outcome of that research for the benefit of society in these developing countries. So ICGB headquarters is in Trieste, Italy, and there are two other centers, one in India, New Delhi, and one in Cape Town, South Africa. And one of the major goals of ICGB is to give more training of different students in these countries, which are mostly developing countries or global south. And in that way, ICGB supports a number of workshops where new students or young generation of scientists are trained in different upcoming techniques. And this was one of the workshops which was partially supported by ICGB, and this is to provide training to young students in a developing country like Peru, which is also part of ICGB.
So malaria molecular surveillance is very important for malaria control. It's important to integrate this molecular genetic data together with clinical data, epidemiological data, but also environmental data in order to inform national strategic planning. The AmpliSeq assay was developed as a multiplex assay, and it can address multiple use cases, which means it can address multiple questions that a national control program could have, and to help it to devise the strategies and also change the allocation of resources, for example.
AmpliSeq is an excellent example of a targeted sequencing technology to very efficiently generate data to understand the biology of malaria, to advance the control of malaria, and maybe to eventually eliminate malaria in countries and regions.
Amplicon sequencing sits at a very important place between science and policy. So for me, this is a very logical focus for a workshop like this. Amplicon sequencing allows you to maximize the innet of your sequencing work to answer focused questions relevant to public health, relevant to policy, but also to tune your methods towards interesting scientific questions. So for me and my group, we seek to take on research that directly informs policy, that can truly help the people we are serving, the people we're working with, the communities where we're active.
So in order for the malaria molecular surveillance to have really an impact on malaria control, it's very important that we create a large mass of human resource capacity. So a large number of people with the capacity to analyze the data, to perform the laboratory protocols, to generate the molecular data, but also to troubleshoot when there's problem arises or we have parasites that don't always perform as we expect them. And that's why it's very important to have a large body of people that have these skills.
But with the increasing number of laboratories that are now generating data, it's going to be important to communicate and to understand how to connect data sets, how to analyze data sets in common ways so that we can still learn from the contrasts we see in data generated in different parts of the world and in different parts of Peru and different parts of Latin America.
And I think in terms of collaboration, we also need to continue strengthening this collaboration, both in data sharing and also to find ways to bring this technology into the hands of the national malaria control programs, because at the end those are is for programmatic questions that needs to be used. In something that we need to put our strength now, it's in better communication with national malaria control programs and seeing how we can better collaborate to really use the AmpliSeq and other malaria molecular surveillance tools to inform policy and practice.
O curso ele tem os tópicos muito aplicados ao cenário de malária no Brasil, que imagine como em outros países, vem como uma proposta de aplicação não só como pesquisa, mas de como implementar. Essa foi uma parte muito importante do curso, de como a gente pode implementar essa ferramenta como uma política pública de saúde num país tão grande como o Brasil.
Mi experiencia en este curso ha sido muy enriquecedora y muy valiosa, debido a que he podido actualizar algunos conceptos que, actualmente ya tenemos nuevas tecnologías, lo que me permite poder tener al tanto estas nuevas tecnologías para poder aplicarlas en mi día a día, en mi trabajo de investigación.
To organize this meeting at the beginning was challenging, but then it was really good because We have a very committed team that can help in different ways and help to do this workshop very well organized. The people are happy. And what was the motivation is because we are working in malaria molecular surveillance in collaboration with the Institute of Tropical Medicine in Antwerp for so many years already. And we think that it was time to share the knowledge we have, the methodology we developed with the other partners around the regions and around the world.
Yo considero que eso es demasiado importante. Es el rol principal de la universidad. No solamente es desarrollar la investigación que puede quedar dentro de la universidad, sino que esa investigación tenga impacto. Entonces yo considero que es trascendental que la universidad participe en forma activa organizando estos encuentros científicos que generan vínculos, un network muy potente y redes colaborativas para mejorar el impacto de la investigación en la población.
We applied to the ICGV to organize this meeting because this has it's a very well-known institute that support developing countries like Peru and other countries that are part from the member states. And also, this fund was complemented with the funds from the Belgian cooperation. This collaboration we have between the Institute of Tropical Medicine, Alexander von Humboldt, and the Institute of Tropical Medicine in Antwerp. And I also want to acknowledge the participation of the National Academy of Science because they also support to organize this workshop.